Good day everyone. Today our group is going to present an experiment with the help of aseptic technique. Now before we further present our experiment, it is important to know about what aseptic technique is. It is in working with the microorganisms, we must have a method of transferring growing organisms from a pure culture to a sterile medium without introducing any unwanted outside contaminants. This process or a method of preventing unwanted microorganisms from gaining access is termed as aseptic technique. Now let's discuss about how we apply or use aseptic technique in an experiment. Materials that are required for our experiment is candle, lighter, test tube holder, and 3 different types of nutrient broth, test tube, stand, and inoculating glue. In order to remove the contaminants for about 10 to 20 seconds, and then we need to heat the test tube mouth that contains contaminated water and put the loop inside it after sterilizing it, and then again heat the test tube mouth in order to avoid any contaminations. Then we need to take up the test tube containing nutrient broth of one type and remove the cap and flame the mouth of it, and also avoid putting the cap of the test tube on the table and hold it in the same hand as inoculating loop to avoid unwanted bacteria. So after heating up the test tube and putting the inoculating loop that contains contaminated bacteria in the nutrient broth test tube and stir it for about 10 seconds, and then sterilize both inoculating loop and the test tube of nutrient broth above the flame to prevent contamination, and close the test tube of nutrient broth by the cap and place it back in the test tube holder. We need to perform the same activity for the 3 times per test tube that contains nutrient broth, and this technique is called as aseptic technique that's we used to follow to prevent contamination while performing any experiment in laboratory. Thank you.